Excuse me, sir. सर आवाज नहीं आ रही।
Sure. The zinc, this is very important metal. Yesterday we started this zinc chemistry, and we talked about its chemistry, that its importance is revealed from the different enzymes which are present inside biological system. This was recognized as essential metal ion inside biological system by scientists. This has many enzymes like lyases, hydrolases, transferases, oxide reductases, isomerases, lycases. The total number of zinc protein is 3,207 inside biological system. We talked about the carboxypeptidase and its role inside the biological system. It plays the role that it hydrolyzes the peptide chain of the proteins. We talked about its mechanism. Then we took the carbonic anhydrase enzyme, which is also very important enzyme, which is converting the carbon dioxide into carbonic acid. Carbonic acid ultimately dissociates into bicarbonate and hydrogen ions. This affects the production of the bicarbonate ion. This is called Bohr Effect, which lowers the pH in the capillaries and decreases the oxygen hemoglobin binding capability. So this effect was called Bohr effect. This is the structure of carbonic anhydrase, and then we talk about its mechanism, how it proceeds to convert the carbon dioxide into bicarbonate ion. Today we'll be starting the chemistry of magnesium, which is also very important metal. Magnesium, this is represented by Mg and is belonging to group 2. It acts as a Lewis acid by accepting electron pairs. Isomerases, lyases, which process carboxylate, are hydroxyl group containing molecule. मतलब ये है कि इसमें ज़्यादातर एंजाइम जिनमें मैग्नीशियम मौजूद होता है, they are called isomerases or lyases, and they process the carboxylate or hydroxyl group containing molecule. It acts as a Lewis acid there, special function in chlorophyll like the hemoglobin. Chlorophyll is present inside plants. We'll be talking about this in detail. ATP must be controlled to Mg2 plus to be utilized. This is involved in Krebs cycle, so it is actually controlling the whole process. We will be talking about this in detail. The first enzyme which contains the magnesium ion, that is xylose isomerase. Xylose isomerase is an enzyme that catalyzes the interconversion of D-xylose and D-xylulose. Xylose are sugars, ठीक है? तो ये एंजाइम जो है ये कैटलाइज करता है the interconversion of xylose to xylulose or xylulose to xylose. interconversion process होता है, ठीक है? मतलब ये एक शुगर को दूसरी फॉर्म में कन्वर्ट करता है, जब दूसरा फॉर्म प्रोड्यूस हो जाए वो नई ज़रूरत ये फिर से वापस इंटरकन्वर्जन करता है. This is very important process. This is called actually this interconversion process is called isomerization. This enzyme belongs to the family of isomerases because it converts the form of the sugar. There is no change in the chemical nature. मतलब chemically तो थोड़े change हो जाते हैं, लेकिन actually the form of the sugar is changed. This enzyme belongs to the family of isomerases, specifically those intramolecular oxidoreductases, interconverting the interconverting aldoses and ketoses. अल्डोज और कीटोस को एक दूसरे में कन्वर्ट करता है, मतलब ये भी ऑक्साइड रिडक्टेज की तरह उनकी तरह काम करता है. Xylose isomerases are also commonly called fructose isomerases due to their ability to interconvert glucose and fructose. ठीक है, fructose isomerase भी इसको कहा जाता है. The systematic name of this enzyme class is D-xylose aldose ketose isomerase. ठीक है. Other names in common use include D-xylose isomerase, D-xylose ketoisomerase, and D-xylose ketol isomerase. ये इसके नाम हैं. This enzyme participates in pentose and glucuronate interconvergence and fructose and mannose metabolism. ये सारे जो हैं ये sugars हैं. Interconversion करता है fructose और mannose या pentose और glucuronate. The most bioavailable sugars according to the International Society of Red Sugars are जो sugars biological cycle में use होते हैं उसको हम bioavailable sugars कहते हैं. So these sugars are glucose, galactose, mannose, fructose, xylose, ribose and L-arabinose. ये sugars biologically available sugars कहते हैं. 20 hexoses and 9 pentoses, including xylulose, were considered to be rare sugars. और इसके अलावा hexoses और pentoses और xylulose ये rare sugars कहलाते हैं. Hence, D-xylose isomerase is used to produce these rare sugars, which have very important application in biology despite the low abundance. तो ये जो enzyme है ये xylose isomerase, actually this is producing rare sugars, which is used in biological systems. Now, the mechanism of action of this enzyme, this is magnesium containing enzyme, which has two magnesium centers. One magnesium center, this is surrounded by two aspartic acid, one glutamic acid, one glutamic acid, and one glutamic acid is bridged between the two magnesium centers. There are two water molecules attached to the magnesium, one magnesium center. This magnesium center is 1, 2, 3, 4, 5, 6 octahedral in structure. The second magnesium, that is having the coordination from glutamic acid to glutamic acid. One glutamic acid is bridged between the two magnesium centers. Two water molecules are attached to it, and it is pentacoordinated. One site is vacant for the coordination. So xylose जो available होता है वो इस xylose जो available होता है, that xylose can be this xylose will actually participate. This xylose will participate in the coordination with this magnesium center, with the second magnesium center. So magnesium coordinates the xylose, the hydroxyl group of the xylose. This is the simplest form of the xylose. वैसे तो ये बहुत बड़ा मॉलिक्यूल होता है, लेकिन यहाँ पे हमने जो है बहुत ऐसे थोड़ा सा छोटा सा उसका लिया हुआ है, पार्ट लिया हुआ है. So this is aldehyde at one side and hydroxyl group. This is connected to the magnesium center. So magnesium 1 and magnesium 2 remain as such. Magnesium 1, then through the reversible reaction, hydrogen is, this hydroxyl group is deprotonated and magnesium produces bond. When it is deprotonated, ये इस magnesium के साथ भी bridging structure बना देते हैं. So that both of these magnesium centers, they are involved in coordination with the xylose. Now, after interconversion, hydrogen which is donated, which is lost during this step, is reabsorbed. And here you may see that this carbonyl group, aldehyde group, this is converted to OH, whereas this hydroxyl group, this is converted to carbonyl. So this cycle, this is actually conversion of xylose to xylulose. The fresh form which is produced, this is called xylulose form. Now another very important enzyme which contains magnesium, that is called isocitrate lyase. Isocitrate lyase, or ICL, is an enzyme in the glyoxylate cycle that catalyzes the cleavage of isocitrate to succinate and glyoxylate. Isocitrate lyase ये एक enzyme का एक class है. ये जो class है, this is very important enzyme which is participating in the glyoxylate cycle. Glyoxylate cycle and Krebs cycle, ये दोनों cycle जो हैं, ये दोनों
स्लाइड शो नहीं हो रहा, सिर्फ ब्लैंक
है,
है
carried out. The glycolate cycle and Krebs cycle both are operating inside biological systems which are producing the energy required for the biological systems. सबके पास स्लाइड शो नहीं हो रहा, या सिर्फ आपके पास स्लाइड शो नहीं हो रहा? Anybody else having anybody else having the problem? The problem is with your internet connection. अगर आपको शो नहीं हो रहा तो इसका मतलब ये है कि आपका इंटरनेट कनेक्शन में मसला है, या आपने जो कनेक्ट किया है वो गलत किया है. फिर से लॉग आउट हो जाएं, फिर से लॉग इन हो जाएं, तो आप शायद ठीक हो जाएंगे. आपको
ओके सर.
Isocitrate lyase, or ICL, this is an enzyme which participates in glycolate cycle. So Krebs cycle, glycolate cycle, these two cycles, they are important cycle and produces energy. And what is the function of isocitrate lyase? It can convert isocitrate to succinate and glycolate. The systematic name of this enzyme class is isocitrate glyoxylate lyase. Succinate forming enzyme इनको भी कहा जाता है. Other names in common use include isocitrase, isocitratase, isocitratase, DS isocitrate glyoxylate lyase, and isocitrate glyoxylate lyase. ये इसके नाम हैं. This enzyme participates in glycolate and decarboxylate metabolism. Glycolate cycle में या decarboxylation processes में ये enzyme participate करता है. This enzyme belongs to the family of lyases, specifically the oxoacid lyases, which cleave carbon-carbon bond. अगर आप इसको कहते हैं the main function of this enzyme, अगर पिछले एंजाइम को देख लें तो वो आइसोमेराइजेशन रिएक्शन में पार्टिसिपेशन कर रहा था. But this enzyme, this is belonging to the class of lyases. Lyases का मतलब है कि this is cleaving something. The cleavage is actually occurring in carbon-carbon bond. ठीक है? So ये जो enzyme है, ये carbon-carbon bond को cleave करता है. Mechanism जो इसका है, you may see this enzyme is involved in the citric acid cycle, like isocitrate ion जो है, this is the isocitrate ion. Here is OH and 3 carboxylic groups. So the enzyme this takes on the hydroxyl group and is deprotonated. Upon deprotonation, this OH is converted to O minus. जो minus group minus sign होता है, this is donated toward the bond, and this bond shifts. So upon shift, the two moieties are produced. One is glyoxylate ion and the other one is succinate ion. So glyoxylate ion and succinate ion This is the main target of this enzyme, isocitrate lyase enzyme. So it happens like this. Now then, another very important enzyme is isocitrate dehydrogenase. असल में ये isocitrate lyase और isocitrate dehydrogenase, these two enzymes, they are involved in the citric acid cycle. ये जो अभी हम बात करेंगे कि साइकिल कैसे प्रोसीड होता है. So these two enzymes, they are involved in this cycle. Isocitrate dehydrogenase is an enzyme that catalyzes the oxidative decarboxylation of isocitrate, producing alpha ketoglutarate and CO2. अब आप देख लें isocitrate dehydrogenase और isocitrate lyase. Isocitrate lyase जो है वो isocitrate को सक्सिनेट और ग्लाइकोलेट में कन्वर्ट कर रहा था, और ये जो एंजाइम है ये ऑक्सीडेशन करता है. और किस चीज़ की ऑक्सीडेशन करता है? ऑक्सीडेटिव डीकार्बोक्सिलेशन. और आइसोसिट्रेट को कन्वर्ट करता है अल्फा कीटोग्लूटरेट और CO2 में कन्वर्ट करता है. सो आइसोसिट्रेट आयन इज कन्वर्टेड टू अल्फा कीटोग्लूटरेट एंड CO2. सो दीज टू एंजाइम, दे आर कंपीटिंग विद ईच अदर. कभी जो है आइसोसिट्रेट डिहाइड्रोजिनेज काम करता है, कभी जो है isocitrate lyase काम करता है. Whenever it is required. This is a two-step process which involves oxidation of isocitrate secondary alcohol to oxalosuccinate, a ketone, followed by the deep carboxylation of the carboxyl group beta to the ketone forming alpha ketoglutarate. मतलब इसमें दो स्टेप्स होते हैं. फर्स्ट स्टेप में क्या होता है? ऑक्सीडेशन होता है आइसोसिट्रेट का, और वो जब ऑक्सीडेशन आइसोसिट्रेट का हो जाता है तो उससे बन जाता है ऑक्सैलोसक्सिनेट, तो कीटोन बन जाता है. जब ये कीटोन बन जाता है तो इस कीटोन की फिर डीकार्बोक्सिलेशन होती है और ये कन्वर्ट होता है अल्फा कीटोग्लूटरेट में. इन ह्यूमंस isocitrate dehydrogenase enzyme or IDH enzyme exists in three forms. IDH3 catalyzes the third step of the citric acid cycle, converting NAD plus to NADH in the mitochondria. Human being में ये three forms में है. एक तो ये catalyze करता है third step को, और उसमें क्या करता है? NAD है, NAD plus को NADH में convert करता है. The isoforms IDH1 and IDH2 catalyze the same reaction outside the context of the citric acid cycle and use NADP plus as a cofactor instead of NAD plus. और जो बाकी दो IDH1 and IDH2 जो हैं वो citric acid के अलावा काम करते हैं, और वो NADP plus को as a cofactor यूज करते हैं, not NAD plus. They localize to the cytosol as well as the mitochondria and peroxisome. Now, the very important thing is ICL competes with the isocitrate dehydrogenase. Isocitrate lyase ICL उसको कहते हैं. ICL competes with IDH. ठीक है? Isocitrate dehydrogenase, an enzyme found in the TCA cycle for isocitrate processing. TCA cycle क्या है? We will be talking about this. The IDH is composed of three subunits, is allosterically regulated and requires an integrated magnesium or manganese ion. ये भी manganese या magnesium की enzyme. ये जो है ये आपका वही isocitrate enzyme है, isocitrate ion है. This is converted to alpha ketoglutarate. अगर आपको याद हो पिछले वाले में isocitrate lyase में this was converted to glycolate, cleavage हो रही थी, सक्सिनेट और glycolate में कन्वर्ट होता है. ये जो है इसमें this is converted, decarboxylation हो गई है, and this is converted to alpha ketoglutarate. Mechanism wise जो है ये 3 steps में show किया गया है कि how this is occurring. The enzyme, this is actually taking part in the decarboxylation of this succinate ion, isocitrate ion, sorry, isocitrate ion. So this is like this. ये next slide में थोड़ा सही show किया गया तो मैं I will take this. Now this is the isocitrate ion. When NAD plus comes, this is converted to NADH plus H ion. Now the H, which is picked up with this NAD plus, this is actually picked up from this hydroxyl group. Okay, so this hydroxyl group is converted to CO. Now another step, this minus charge, this is shifting its charge to this bond. As a result, this bond is shifted over here, and when H comes, so this is converted to CH2, and this whole moiety is released as CO2. So alpha ketoglutarate and CO2 are released during isocitrate dehydrogenase reaction. ठीक है? Citric acid cycle और Krebs cycle, ये दो एंजाइम्स हम पढ़ रहे थे कि ये सिट्रिक एसिड या क्रेब्स साइकिल में पार्टिसिपेट करते हैं. क्रेब्स साइकिल आप लोगों ने पहले भी पढ़ा हुआ है. क्रेब्स साइकिल इज अ सीरीज ऑफ chemical reaction used by all aerobic organisms to release stored energy through the oxidation of acetyl CoA derived from carbohydrates, fats and proteins. ठीक है, एनर्जी रिलीज करता है, एरोबिक साइकिल है, और इसमें all form of the metals like जो हम कहते हैं this is carbohydrates, fats, इनके oxidation process से होते हैं. In addition, the cycle provides precursors of certain amino acids as well as the reducing agent NADH that are used in numerous other reactions. मतलब इसका ये काम सिर्फ नहीं है कि ये एनर्जी देता है, इसके अलावा भी इसके फंक्शंस में आते हैं कि ये अमीनो एसिड्स प्रोडक्शन भी करता है, इसके अलावा रिड्यूसिंग एजेंट लाइक NADH प्रोडक्शन भी करता है. और वो दोनों आपको पता है कि अमीनो एसिड्स आर यूज्ड फॉर द प्रोटीन एंड NADH दे आर यूज्ड फॉर अदर साइकिल्स एज वेल. At central importance to many biochemical pathways suggests that it was one of the earliest components of metabolism and may have originated abiogenically. ठीक है, ये बहुत पहले का साइकिल है जो एनर्जी के लिए बायोलॉजिकल सिस्टम यूज कर रहे थे. The name of this metabolic pathway is derived from the citric acid, a tricarboxylic acid often called citrate, as the ionized form predominates at biological pH 6. ठीक है, इसमें ज़्यादातर ये सिट्रेट आयन जो हैं ये ज़्यादातर डोमिनेट करते हैं, तो इसलिए इसको सिट्रिक एसिड भी कहा जाता है, सिट्रिक एसिड साइकिल भी कहा जाता है. That is consumed and then regenerated by this sequence of action to complete the cycle. The cycle consumes acetate in the form of acetyl CoA and water, reduces NAD plus to NADH. एसिटेट आयंस एसिटाइल को एंजाइम ए जो है इसकी मौजूदगी में कन्वर्ट होता है NAD प्लस टू NADH, रिलीजिंग कार्बन डाइऑक्साइड. कार्बन डाइऑक्साइड जो रिलीज हो जाता है सेल्स के अंदर वो फिर हेमोग्लोबिन कैरी करता है. NADH regenerated by the citric acid cycle is fed into the oxidative phosphorylation electron transport chain में NADH जो है यूज होता है. The net result of these two closely linked pathways is the oxidation of nutrients to produce usable chemical energy in the form of ATP. ATP is produced होते हैं. This is the cycle. अगर आप इसमें देख लें, this is pyruvic acid is converted to acetyl CoA, then acetyl CoA is converted to acetic acid, which is converted to citric acid. Now this citric acid is converted to citric acid, which is done by the enzymes, and this is citric acid. This may be directly converted to alpha ketoglutarate acid, which is again the function of the enzyme. So you may see that this conversion is done by the magnesium enzymes. अगर ये काम ना हो, so you will not get the whole Krebs cycle. So the Krebs cycle is dominated. This is actually controlled by this magnesium enzyme, magnesium containing enzymes. The alpha ketoglutarate acid is converted to succinic acid, and then fumaric acid, and then malic acid or इसमें हर स्टेप में FADH2 NADH और एनर्जी रिलीज होती है। ओवरऑल जो इस साइकिल में एनर्जी रिलीज होती है वो आप लोगों को पता है कि वो कितनी ATPs रिलीज करता है इस ओवरऑल प्रोसेस में। We are not talking about the Krebs cycle in detail because this is not not in our course. I was just only checking this Krebs cycle in order to show that citric acid, isocitric acid, and alpha butyric acid किस तरीके से इस साइकिल में इन्वॉल्व हैं and how they are converted to, मतलब citric acid is converted to alpha ketoglutaric acid by these enzymes. ठीक है, मतलब ये जो दो enzymes function कर रहे थे तो ये किस तरीके से इनको convert कर रहे थे. If you people have any question regarding this topic, you may ask.